Hi everybody, my name is Charlotte Minio, and I'm an undergraduate student studying biochemistry at Union College. And this summer I'm going to be working with a series of enzymes called metacaspases. So metacaspases are proteases, which means that they're proteins that break apart other proteins. And they're involved in a process called programmed cell death. So programmed cell death is pretty much a very organized, methodical way for cells to kill themselves. And in multicellular organisms, killing off some cells is a very natural and beneficial process for the whole organism. So we're interested in understanding how that happens, as well as using this pathway, using programmed cell death, to develop a new kind of antifungal drug. So essentially, by understanding the metacaspases a lot better, you could potentially design a new kind of antifungal that would work by activating the metacaspases, and then once the metacaspases were active and they were breaking apart the proteins that they need to break, that would induce programmed cell death, so you'd have dying cells, which is exactly what you want in an antifungal drug. You want to reduce the number of fungal cells that you have. So this is really important because all sorts of human diseases, from ringworm to athlete's foot to really serious illnesses like sleeping sickness, can be caused by fungi. And in recent years, more and more antifungal drugs have been prescribed, so more and more fungi have become resistant to antifungals. Just like you hear about antibiotic resistance, you also have antifungal resistance. And so in the coming years, as current antifungals become less and less effective, it's important to have new drugs that can be offered to patients to help cure their illnesses. But before all that can happen, we need to understand the metacaspases a lot better. So pretty much what my lab is doing is studying what factors change the metacaspases, change their activity, and determine when and where they're active. So pretty much we're working on figuring out what kinds of proteins do the metacaspases cleave, and then where do they break those proteins apart. And then we're also working on figuring out what makes them active. So just because we have a metacaspase and we have the protein that it's supposed to cleave, doesn't necessarily mean that that process is going to happen. So that's how in a cell, you don't have cells randomly dying all the time from programmed cell death. There's specific signals that pretty much tell the metacaspase, okay, it's time to undergo programmed cell death. It's time to do your thing and break apart these proteins. So one chemical messenger that's pretty common as an activator for enzymes is calcium. And so my lab is studying over the summer how calcium can change the activity of our metacaspases. So pretty much what we're going to do is we're going to take a sequence of DNA that codes for the metacaspases. If you remember the central dogma of molecular biology, you have DNA that codes for RNA, which is another kind of nucleic acid. And then the message in that RNA tells ribosomes what kinds of amino acids to put in what order. And then you have pretty much the precursor to your protein, which then folds up and it becomes an active protein. So what we're going to do is change the sequence of nucleotides in our DNA. We're going to mutate it so that it codes for different proteins, codes for different amino acids, which will change our protein. So once we do that, we're going to take that DNA and we're going to put it into some E. coli. We're going to pretty much use the E. coli as a little factory to produce protein for us. So we're going to let those E. coli grow, let them produce our metacaspases, and then once we have this protein, it's mixed up with all sorts of other junk from our E. coli. Lots of other proteins, lipids, carbohydrates, etc., etc. So what we're going to do is we're going to purify a cell extract from our E. coli. We're pretty much going to break them apart, purify our protein so that all we have left is the metacaspase that we want to work with. And then we're going to see how our mutated metacaspase compares with normal, unmutated, wild-type metacaspase. So the way we'll do that is we have these special little, very short proteins, and when they're broken apart, they produce light. And it's not visible light, it's fluorescent light. And so what we can do is we can use these special machines called plate readers that will measure changes in the amounts of light produced, and we can compare. So it's pretty much going to tell us how active the enzyme is. So the more light that's produced, that means that the enzyme is breaking apart more proteins and it's more active. So what we'll be able to do is compare our mutated enzyme and our wild type enzyme at some different concentrations of calcium to see how active the enzyme is. So what that's going to tell us is, are the enzymes dependent on calcium? If our mutated enzyme is no longer producing light, is no longer cleaving other proteins, then we know that calcium is really important for the activation of this enzyme, and drugs targeting the calcium binding site could be really useful targets for developing new antifungals. Thanks for listening.